Good morning, Vanshika. Good evening, Vanshika. Sorry. Okay. Yes, I'm Rahul Saha, and we'll be completing this chapter right now. The sexual reproduction in the flowering plants. Okay. In the last class, we had discussed regarding. Good evening, Aman. So we had discussed regarding the different types of structures, like of the structure of the maize seed or maize grain, then the structure of the castor seed, the hypocotyle, the testa, the tegmen, the endosperm, and all that. So in today's class, we'll be discussing firstly the phenotype of a fruit, then dormancy, then apomixis, and then we'll be switching over to oogenesis of the next chapter. Okay. So coming on to the fruit. So fruit may be defined as what? By definition, you should be saying that fruit may be defined as a mature and ripened ovary. And ripened ovary. The stimulus of fertilization, you can say it not only develops ovules into seeds, but also brings about other changes, other morphological changes in the flower, you can say. So usually all the parts of a flower, except the ovary, what happens is they wither away, they degenerate. But the ovary begins to enlarge simultaneously with the development of seed, and then development of seed happens, ovary enlarges, and later on what happens is we have the ultimate formation of, or you can say the formation of fruit 2. The formation of fruit. Now a true fruit is one which develops from a single ovary of a single flower with no other part outside of the ovary. That is known as a true fruit. You should be knowing this very, very clearly for any kind of exam, whether board or neat, that a true fruit is one which develops from a single ovary. From a single ovary. Okay. And then of a single flower with no other part outside the ovary. Like for example, I can take the name of Mangifera indica, mango, then simply tomato. Okay, these are the true fruits, you can say. So this is the proper definition of a true fruit, which develops from a single ovary of a single flower with no other part developing from any part outside the ovary. Okay. Now, when does a fruit become false? False fruit means it is also known as a spurious fruit or a pseudocarp. Okay. So a false fruit or spurious slash pseudocarp is a fruit when, you can say, other floral parts, other floral parts also take part in the formation of fruit. Also take part in the formation of fruit. Okay, like you have lychee, you have apple, you have pear, etc. Right? So what happens is all aggregate and multiple fruits can be considered as false fruits. So you might have heard of aggregate fruit, you might have heard of multiple fruits. So all the aggregate and multiple fruits can be considered as a false fruit. Directly in comparative examination, you can write that. You can tick on false fruit if you're asked about any aggregate or multiple fruits. Okay, so one more thing I'll tell you, the fruits which result from the inferior ovaries are false, as the wall of such an ovary will do what? It has a part of thalamus fused with it. Very, very important for comparative examination, this line. So, the fruits resulting from inferior ovaries. Inferior ovaries, okay, are false, as the wall of such an ovary, you can say, has a part of thalamus fused with it. Fused with it. Now, you can give the example of mulberry, even pineapple. These are also false fruits because they emanate from the inferior ovaries. Okay. Now, fruits of also, you can say apple, pear. Okay. Again, a good comparative question. Fruits of apple, then you can say pear, then you can say strawberry, then you can say cashew, right? They are also false. Why? Because the edible part is mainly the fleshy thalamus. Yes, the edible part is the fleshy thalamus. Next, brinjal is also a false fruit as it has, you can say, persistent calyx. Okay, brinjal is also considered a false fruit due to consisting persistent calyx. Very, very important. These important, you can say, these specific pointers are extremely significant for your board and NEET both, right? 1 mark question. Now, coming on to dormancy, right? So, if seeds germinated at the earliest times, okay, as per the, you can say, conducivity or the permittivity of the conditions, many would begin growth before the onset of winter. And then what would happen? They would find themselves killed by an unfavorable climate because they had not developed completely enough to survive. So, in order to circumvent this specific problem, what happens is the evolution has put a natural selection, and that natural selection has led to the effect or to the onset of dormancy. So for this reason, many seeds undergo, you can say, a period of dormancy before germinating. A period of dormancy before germinating. Okay. Now, sometimes a particular set of conditions they need to be satisfied before the seeds can begin the growth again. Now, this period of inactivity is known as dormancy. Inactivity means the metabolic growth is almost completely ceased. All the anabolic and the catabolic pathways, the biochemical pathways, are extremely slowed down. Many are completely ceased. Only you can say, minimal amount of ATP is produced, minimal amount of starch is being used up, right? That is known as the period of inactivity, is called dormancy. Now, what kind of periods can induce dormancy? Okay, so you can say a period of sustained cold that can induce dormancy. Sustained cold. Next, a set period of time, you can say, which will also allow certain extremely slow chemical process to occur. Then you can say a certain amount of light above a given intensity, high intensity of light. Then you can say partial breakdown if happens of the seed coat, which is the testa. Okay, so that could be the problem. The heat of a flash fire, anything can cause dormancy to begin. Okay, so these are the things that pertain to the dormancy of a seed, right? Then coming on to the next topic, apomixis. Okay, so now if you talk about apomixis, so the apomixis is the formation of new individuals. Okay, apomixis is the formation of new individuals through asexual reproduction. You can say without involving the formation and fusion of gametes. That is known as apomixis. Okay, so you can say this is very close to your parthenocarpy, or sorry, to your parthenogenesis, right? So what you can say is, you can say, differentiate between apomixis and, you can say, parthenogenesis. You can really differentiate. Okay, so once you differentiate, so what you can get is, I'll give you the points regarding the differentiation. So they are very critical. Okay, for your board examination, direct question, one of those question comes, right? What is the difference between an apomixis, you can say, and parthenogenesis? So you can say that apomixis is the formation of apomixis is the formation of the seed without meiosis and syngamy. That is the in-depth explanation of the seed without meiosis and syngamy. Now, there is one more thing, you can say amphimixis. Okay, now if you talk about amphimixis, that is also there in your course. Amphimixis is the sexual reproduction. It is the sexual reproduction You can say where the male gamete fuses male gamete fuses, okay, with the female gamete to form the zygote. To form zygote. Okay. Then we can come to, you can say, parthenogenesis. Now, what is parthenogenesis really? Parthenogenesis means the formation of embryo from an unfertilized egg or female gamete. Parthenogenesis is nothing but the formation of embryo from an unfertilized egg. But meiosis has occurred. Their meiosis did not occur. Here from an unfertilized egg or female gamete. So that is the only slightest of the difference is present between, you can say, apomixis, parthenogenesis, and one more thing I included, that is the amphimixis. Okay. So you can say that apomixis is a form of asexual reproduction. You can say that mimics sexual reproduction. So what you can say is there are other terms also. If there is a term known as agamospermy, okay, that is also in your course, agamospermy. So if we talk about agamospermy, that is the formation of new individuals, okay, new individuals by asexual reproduction without involving fusion and formation of gametes. Without involving fusion and formation of gametes. Another, you can say, subtype, formation of gametes. Now, if you want to define, or you can, if you want to segregate agamospermy, it can be of the following types. Okay. Now, just now you will see how I can relate the examples and make it completely crystal clear. Types of agamospermy, you can say. So types of agamospermy means number one, it will be adventive embryony. Here what happens in this case, you can say one or you can say few extra embryos. One or few extra embryos are are formed, you can say, from a diploid cell of nucellus or integument but never from the egg cell, from the egg itself. Okay, so you have the example of mango, opuntia, okay, here that comes under the adventive embryony. In the second one, you can have recurrent apomixis or recurrent agamospermy. So type 2 is recurrent apomixis. In this type, what would happen is a diploid embryo sac. Here, a diploid embryo sac is formed which has a diploid egg. Which has a diploid egg. Okay, it is also known as oosphere. Now the diploid egg grows parthenogenetically into diploid embryo. The diploid egg grows. There is no fusion happening. See, the diploid egg grows parthenogenetically. Parthenogenetically into a diploid embryo. Into a diploid embryo. So this has been seen in case of species, you can say genus Allium. Okay. So this is known as recurrent apomixis, that a diploid embryo sac is formed which has a diploid egg directly without fusion, okay, of the male gamete, and then the diploid egg would grow parthenogenetically because it is always remember it is agamospermy. Okay, no sperm would be present. Okay. Next is non-recurrent, that is recurrent apomixis, or you can say recurrent agamospermy. Next will be non-recurrent agamospermy, type 3. Non-recurrent agamospermy. What would happen in this case is it is the development of the embryo. It involves the development of the embryo, okay, from haploid female gamete without fertilization. Okay, means the egg without fertilization. Okay, very specific thing. It is also known as simply parthenogenesis. This I can call it as parthenogenesis also. Okay, a type of parthenogenesis. And number 4 could be direct parthenogenesis. Okay, which will have almost the same definition. Okay, parthenogenesis, that is, it is the development of seed or embryo, development of seed slash embryo from a female gamete without fertilization. So they all come under, you will be noticing the relation that apomixis, parthenogenesis, they all come under, you can say, agamospermy. Okay. Then you can say parthenogamy. Okay. One could be parthenogamy. Just variations of agamospermy. That's all. Now, parthenogamy means it is the union of two incompatible gametes. very very special thing, unique stuff. It is the union of two incompatible gametes. Meaning that fusion of two female gametes, not male and female, fusion of two female gametes, very important for the from the NEET perspective or competitive exam perspective. Fusion of two female gametes. That is known as parthenogamy. Okay, so this is parthenogamy. Then you might have learnt about, I had taught you about apospory. Okay, so that will also come under agamospermy only, the apospory. It involves the development of gametophyte from directly the sporophyte. So I'm just revising it for you. Formation of gametophyte directly from the sporophyte. Sporophyte. That is apospory. Spore means the formation of the gametophyte. Okay. From the sporophyte. Okay. Apogamy. That is the formation or development of a sporophyte from the gametophyte. But the rule will be without the fusion of gametes. Without the fusion of gametes. So these things you already know, but I'm repositioning it in one, you can say under one category, so that it becomes really, really easy for you in the long run, that you have a specific category known as agamospermy. Under that you can classify all these different types of agamospermy. Then one more thing you can have is the last thing, diplospory. Now in this case, diploid embryo sac, the diploid embryo sac can develop directly from the diploid megaspore mother cell. From the diploid megaspore mother cell. Okay, so that is known as diplospory. So these are the events that you should be keeping in your mind with utmost, you can say, sincerity. Once you go for, good evening Asma, once you go for, you can say, the diplospory event. Okay, so now what we will be doing is we will be going for the next chapter. Okay, where we will be starting the oogenesis directly, and then we'll be coming on to the different male and female reproductive tract. Okay, so first I'll give you a specific, you can say, table where you will be finding age, then follicular histology, then meiotic events in ovum, and also the chromosomal complement, where how many chromosomes are present you will be looking at. Okay, so the first thing is, you can say, you can make a table where you will be having age. Okay, you will be having follicular histology. Okay, Then meiotic events in ovum. Then you can have also the chromosomal complement, means how many DNA and how many chromosomes are present. Complement. You can just differentiate like this. Now, if we talk about the fetal period, okay, when you are still developing as a fetus in the mother's womb or embryo, when we consider oogenesis, so first is the fetal period. Under fetal period, there wouldn't be any formation of follicles. So there wouldn't be any follicles. So I can say no follicle. Okay. Then meiotic events in ovum, that is known as, it will be still known as oogonium. Okay. Then here we have DNA, which is 2N, chromosome which is, you can say chromosome 2N, DNA 2C, double and double. Okay. Now before or at birth, before or at birth, if you talk about this, the follicular histology, the follicle will be a primordial follicle. We'll describe everything in detail, don't worry. Just note down this table first. Primordial follicle. Okay. Then we'll be having primary oocyte. Okay. So here we had mitosis oogonium. Oogonium became differentiated into primary oocyte. Okay. Mitosis, then primary oocyte. Then now the chromosome is still same, but the DNA has doubled, means it has become 4C. Chromosome is diploid, DNA has become 4C. Now if we come further down the lane, we come after birth. After birth, what happens is the primordial follicle becomes primary follicle. Okay, it becomes directly the primary follicle. Then the primary oocyte would just initiate, you can say, meiotic event, and it would have entered meiosis, but it will still stay as primary oocyte. Okay, so just meiosis would begin, it would still stay as primary oocyte, and then what would happen, it would get arrested also in the diplotene stage. Okay, in the diplotene stage of prophase 1. How that arrest would occur, I'll just show it to you. Okay, so again 2N 4C. If you come further down the lane, we can come to after puberty now. The event is now after you have reached the event of puberty, sexual maturity. That means the primary follicle would have now become a secondary follicle. All these things will be described in detail. Okay. Now secondary follicle, okay, which was already arrested in the diplotene stage here. It was arrested, the primary follicle was arrested in diplotene stage. That arrest would be broken. Okay, that arrest would be broken, and then what you would become is, you would again complete your divisions and become a fully mature primary oocyte. Okay, primary oocyte fully mature. Here again you will be 2n4c. 2n4c. Okay. Quadruple DNA, double chromosome. Then next up is after puberty only, there is no further event. Now the secondary follicle would become tertiary follicle. Okay. Now this will become tertiary follicle. When it becomes tertiary follicle, the first meiotic division would have been completed here. Meiosis 1 complete. And when complete the first meiotic division is complete, start of second meiotic division, what you will be having is you will be having secondary oocyte plus polar body 1. Right, so secondary oocyte plus polar body 1. And then here you will be having now division of the cell. That means you are becoming now 1n2c. Division has happened, so 1n2c. Now next event is you can go for now LH search ovulation. Okay, and go for ovulation. Then I can call it an ovulated ovum. Okay, ovulated ovum. Now it is an ovulated ovum. Okay, it would still be in the secondary oocyte first polar body stage and arrested in the metaphase 2 of meiosis 2. Secondary oocyte plus polar body 1 arrested at metaphase 2 of meiosis 2. Metaphase 2, right? Now again it is 1 and 2c. Now next is if fertilization does happen, means sperm does get in, that is the fertilized ovum. Fertilized ovum, and in this case the fertilized ovum would break the, you can say, meiotic arrest at metaphase 2 and second polar body will be produced. So arrest would be broken and then this will be a fertilized ovum and second polar body. Yes, second polar body. Okay, it will be now 1n1c. Okay, plus sperm. Means our sperm has come in plus sperm. Okay, that is also 1n1c. In the end, the zygote will become 2n2c in the end. So that is known as becoming a fertilized ovum, or fertilization now does take place. So this is the summary in a way that will help you a lot. Okay, that will help you a lot regarding the, you can say, comprehension and decipherability of the entire, you can say, meiotic events which are related to, you can say, oogenesis pathway. Now what we can do is we can show you, okay, I can show you the specific oogenesis stage. Okay, now in oogenesis what happens if we go deep into oogenesis? Okay, what happens is, let's now study oogenesis from depth. Okay, from the conceptual point of view. So you can say of the approximately there are 2 million primary oocytes which are present at birth. Okay, understand it very very carefully. 2 million, not 1 or 2, 2 million primary oocytes are present in the ovaries at birth. Are present the ovaries at birth. Okay, so that is how many ovaries are present at birth, you can say. Now what happens is only about 40,000, okay, only about 40,000 ovaries, sorry, only about 40,000 primary oocytes are arrested in the diplotene stage of the first meiotic division. Okay, so that is there. So what you have is only about 40,000, okay, of which are arrested in the diplotene stage of the first meiotic division, they can survive until puberty. So out of 2 million, how many are able to survive? Only 40,000 are able to survive. So 40,000, you can say, primary oocytes can survive, and 2 million are arrested, but 40,000 are able to survive, which have been arrested in the diplotene stage. Okay, so now only 40,000 survive. Okay, so that is the case. Next is, from this number, approximately 400, that is one per menstrual cycle, are actually ovulated in the entire life. Okay, the rest of the primary oocytes of the 40,000, they degenerate without leaving the ovary. Okay, they degenerate, the rest degenerate without even leaving the ovary. Okay, they degenerate without even leaving the ovary, but many of the 40,000, they undergo some, you can say, further development before becoming dead, or before reaching a stage known as degeneracy, or it is known as atresia. They become atretic. Okay, non-functional, dysfunctional, they become atretic. Okay, so they reach further developmental stages, but they cannot go for ovulation. Okay, those primary oocytes, they cannot go for ovulation. Make it very, very clear. Now the egg, along with its surrounding cells, is called a follicle. Okay, what happens is, you can say, maturation of the egg is intimately bound with the development of its cellular covering. Know this very, very carefully. Because of this, considering the development of the egg and its surrounding follicular cells, because of this only I had given you that specific table. Okay, now that table would be making more sense to you. Because of this only I had explained to you, or I had given you that specific table. Okay, so what happens is, the growth period for a human oocyte, okay, lasts between 110 to 120 days. So human oocyte growth period lasts between, you can say, 110 to 120 days. Then during its period of growth and maturation, the oocyte must amass in its cytoplasm, must be able to, you can say, accumulate in its cytoplasm all kinds and amounts of organelles and materials required to carry, or required to sustain, you can say, the embryo through the first few days of its existence. Now, these materials will include what? These materials would include gene products, okay, which are transcription and regulators, okay, or transcription factors, you can say. Transcription factors which are needed in the eukaryotes, okay, transcription factors which are extremely needed, they are, you can say, indispensable to guide the early cleavage in the embryonic stages or during the first stage of embryogenesis. The cleavage stage we will talk about later on. Okay, so that is the one of the most important things, okay, which happen in the growth and maturation phase of the oocyte. So in the embryo, if you see, the oogonia are completely naked, but after when meiosis begins in the embryo, the oogonia are completely naked. They are not covered by any granulosa cells, follicular cells. But after the meiosis begins, okay, cells from the ovary, they partially surround the primary oocytes to form the primordial follicles, you can say. Then they are surrounded partially, okay, by the cells of the ovary itself, okay, to make them primordial follicle. Okay. Now this is known as a primordial follicle. And when you reach the stage of birth, so by birth the primary oocytes, you can say, are completely covered or are completely invested with a complete layer of follicular cells. Okay, they are known as the granulosa cells. They are layered, they are covered by a complete layer of follicular cells, and the complex of primary oocyte and these granulosa or the follicular cells are then called as the primary follicle, which I had already given you or shown you in the table. Okay, so then when you reach the stage of birth, they are completely surrounded by you can say by one layer of granulosa or follicular cells. Now they are known as this combination of primary oocyte and follicular cells, one layer thick, which are surrounding the primary oocyte. Now they are known as primary follicle. Now this structure is known as primary follicle. Okay. Now, while the primary follicle takes shape, okay, a prominent translucent non-cellular material, it is very very important, a prominent translucent non-cellular membrane, you can say, which is known as zona pellucida, forms between the primary oocyte and its enveloping follicular cells. Very very important, meaning that if this is the, you can say, primary oocyte, so you will form a zona pellucida, you can say this will be the zona pellucida, and then you will be forming the, you can say, one layer, you can say this is the granulosa cell. Okay, so this layer is the zona pellucida. Now the zona pellucida is a translucent and non-cellular membrane, very very important, translucent and non-cellular membrane. Okay, it is made up of, you can say, various glycoproteins and glycosaminoglycans. Okay, meaning that glycosaminoglycans are, you can say, specialized sugars, disaccharides which have specified sulfur group, okay, and amine group, like heparan sulfate, keratan sulfate, dermatan sulfate. Okay, then you have hyaluronic acid. Okay, so these are known as the glycosaminoglycans. Okay, the heparin is nothing but a hypersulfated form of heparan sulfate. It's a glycosaminoglycan, specialized disaccharides. Okay, now they also are responsible for being the sperm receptor. You have ZP1, ZP2, ZP3. Okay, they are the sperm receptors also which bind with the sperm. So in case of rodents, what happens is the components of zona pellucida, they have four glycoproteins and glycosaminoglycans are synthesized almost entirely by the developing egg itself in the rodents. But in other mammals, the follicular cells also contribute towards the synthesis of the zona pellucida. Means in humans, both the developing egg and the granulosa cells or primary follicular cells, they contribute towards the development, you can say, development of zona pellucida. So evolutionarily it does contain sperm receptor. Okay, so it is an extremely pivotal, you can say, entity, non-cellular entity present. Okay, so it's a very important, you can say, entity when it comes to the oogenesis or the pathway going for the fertilization. Now the oocytes, okay, and its surrounding follicular cells, they maintain an intimate structural and functional relationship, you can say. They have two types of projections or connections. One type is known as, it is mediated by effectin, okay, which means that it connects with the microvilli projecting from the oocyte through the, you can say, gap junctions. They have specialized gap junctions, and what they do is they will cause the flow of material from the granulosa cells to the oocyte. The other type is rich in microtubules, okay, which forms the adherence junctions at the, you can say, invaginations of the surface of the oocyte. Okay, so one will be your gap junction, another will be your adherence junction, and this adherence junction is perpetuated by microtubule alpha beta tubulin dimers. This will be perpetuated by the F actin, filamentous actin, or polymer of globular G actin. Okay, that is the microfilament. Know it completely. You might have read about the extracellular proteins, okay, which are extremely responsible for the, you can say, the adherence of the cell. How do the cells adhere to each other? You might have read about tight junction, occluding Claudine, gap junction, then adherence junctions, right? Then desmosomes, hemidesmosomes, and all that. So this will keep on transporting material from the, you can say, the granulosa cells to the developing egg. Okay, so this keeps on occurring. Now what happens is the meiotic arrest at diplotene stage. Okay, the meiotic arrest at diplotene stage is perpetuated by these events only. What happens is there is a specific event known as the formation of cyclic AMP, okay, which we'll discuss in detail in the next class. That cyclic AMP only keeps the diplotene arrest active, keeps, or you can say sustains the diplotene arrest until the puberty happens, until the onset of puberty. Okay, and these gap junctions, they play a pivotal role in maintaining the concentration of cyclic AMP and also inhibiting an enzyme known as PD3A, phosphodiesterase 3A, because that would degrade CAMP into 5' AMP, and therefore a diplotene X would break down. How it will break and how this PD3A is inhibited, I'll explain to you in the next class, that how does the diplotene stage, or how is the diplotene stage, or the How is the diplotene arrest maintained from birth till the onset of puberty? Okay. So I hope you were able to comprehend the lecture, and if you have any kind of queries or doubts, you can definitely contact me on my personal number. Okay, I'll be replying as soon as possible. Okay, then thanks a lot. Let's meet you in the next class.